Hello, ladies. Today we're going to be talking about evolution as genetic change and what that means. So, today's objectives that we're going to be looking at, you will examine how natural selection affects single gene and polygenic traits, and you are also going to analyze what genetic drift is. So, first and foremost, before we start out, make sure you remember what fitness is, and that is the ability of an organism to survive and reproduce in its environment. So, the more fit it is, the more able it is to survive and reproduce. The less fit it is, they're probably going to be dying. So, when we talk about natural selection on a single gene trait, it's going to be able to lead to allele frequency changes. And when we have a change in the allele frequency, we're also going to see a change in phenotype frequencies. Remember, when we talk about evolution and genetics, a change in the allele frequency of a population is what evolution is. So, an example of this is if we see lizards and we talk about red being an easily seen color, brown being decently camouflaged, black being the best camouflage, we can look at the graph on this next slide. And what we can see is that the initial population has 80% brown, 10% red, and 10% black. Well, the red's going to die out first, so generation number 10 here, we see 80% brown, 20% black. And then slowly over time, we're going to see a shift to favor the black lizard. You can also think back to the polar bears here and how slowly over time, there's going to be a shift to favor white bears versus brown bears. So remember, when we talk about natural selection and polygenic traits are going to be shown on a distribution curve that's normal distribution, or that bell curve that we looked at from before. So remember, polygenic traits are controlled by multiple genes, and therefore we're going to have a lot of different phenotypes. So when we talk about natural selection and how it acts on polygenic traits, it's going to affect the relative fitness of phenotypes. And when you talk about affecting relative fitness, you're also going to end up producing one of three different types of selections. And we're going to look at those in depth. We have directional selection, stabilizing direction, or disruptive selection. So the first one we're going to talk about is directional selection. And this is when one individual's at an end of the curve have a higher fitness than individuals in the middle or the other end. So you're talking about one of the extremes is going to have a better fitness. And literally what we're going to see is that range of phenotypes shift because of the success at surviving and reproducing. So Darwin's finches were a great example of this. So let's say you're on an island where the only food present are large seeds. So if there are only large seeds on that island, the birds would therefore need larger beaks to survive. So instead of the average being a middle-sized beak or a medium-sized beak, it's going to shift to the larger-sized beak. So if we look at the picture on the next slide, the very first one, we see here that in directional, that very top graphic, the dotted line represents the original distribution of the bell curve. So in directional selection, if we're talking about large beaks being more favorable, we literally see a shift in where the averages are and where the distribution is. So next is stabilizing selection. And this is where individuals near the center of the curve are going to have a higher fitness than individuals at either end. So essentially what this means is that the center of the curve is going to be a lot larger. So think of this babies being born. So if a mom has too big of a baby during birth, it's going to make birth really painful, uncomfortable, can have complications involved. And if their baby is too small, then it can lead to complications as well when you talk about the development of the baby. So really, when we talk about birth in humans, natural selection is going to favor medium-sized babies. And this is where we get this stabilizing selection curve that we see on the next slide. So here in the middle, we see stabilizing selection. Again, the dotted line represents the original distribution, and we see that that middle solid line is going to represent the more likely to survive, and it definitely favors those in the middle. You can see the two ends are coming in, the middle is extended up. So the last one we're going to talk about is disruptive selection. And this is when individuals at the outer ends, so both ends, have a higher fitness than those near the middle. So this type of selection acts against the intermediate individual. So if you're medium or you're right in the middle, you're not going to be well suited to survive compared to the ones at the end. And actually with disruptive selection, if there is enough pressure, it could cause the curves to split and create two distinct phenotypes. So again, think of the finches. What happens if there's only small seeds available and only large seeds? The small beak birds and the large beak birds are going to dominate and outcompete the medium beaked birds. And that's when we see the selection graph we have on the next page. So at the bottom here we have disruptive selection, and you can start to see that the intermediate, the numbers are going down, and instead there are two peaks we see with disruptive selection that favor either end, or both ends, in this situation, of the chart. So we can start talking about genetic drift as well when we talk about all this. And essentially, natural selection is not the only source for evolutionary change at all. An allele can become more or less common simply by chance. And that random change in allele frequency is called genetic drift. And we see genetic drift occur in a lot of different ways. So the first way we're going to look at is what's called the bottleneck effect. And this is going to be a change of frequencies in alleles following a dramatic reduction in the size of the population. So essentially what I want you to think of is a giant disease or disaster that comes and affects this population. So you can think of the zombie apocalypse, tornadoes, earthquakes, volcanoes, fires, massive eruptions, whatever. And essentially, the population is going to shrink to a very small number. And when you talk about bottlenecks, they can sharply reduce a population's genetic diversity. So this whole disaster of whatever magnitude comes and kills a lot of the individuals in a population. So when you look at the smaller population's gene pool, it's probably going to have a different allele frequency from the original population we had. So if this small population is able to survive and grow, the alleles are going to be very different in frequency compared to the original population. Then we have what's called the founder effect. And allele frequencies are going to change as a result of the migration of a small subgroup of a population. So essentially, when people go out and move to different areas, they're migrating in some way. And they are going to spread their gene pool into another gene pool. And this can affect how that population grows. So a few individuals that colonize in a new habitat are going to continue, grow, develop in that new habitat, and we're going to see these new allele frequencies begin to change depending on where they live. And the great example is on the next page with these beetles. So we have a sample of the original population, that big diverse group. Well, two smaller groups go out and let's say they move, they get on a ship with some humans and they inhabit two islands that don't have any beetles on it. Well, these populations are going to grow on their respective islands, both founding population A and B, and we can see what the descendants of both islands are going to look like. And if we compare each individual island to the original population, it's very different than the original. So we do see a shift when it comes to allele frequencies. So when you talk about allele frequencies and all this, you have to talk a little bit about genetic equilibrium. And essentially, allele frequencies are unchanged and they're constant in genetic equilibrium. So essentially, this is done by calculating a hypothetical population. And if the current population does not meet the hypothetical frequencies, then the population is evolving. So even through sexual reproduction, a species can remain in equilibrium. It just all depends on whether or not they meet this hypothetical population frequency. So that's when we get to this big complicated equation known as the Hardy-Weinberg principle. And essentially this makes predictions of allele frequencies for the next generation. It's really similar to Punnett squares and how they work, except with instead of individuals, it works for an entire population. So I hope you all are ready to do some math, because this is going to be the chapter in the section where we see quite a bit of math come into play. So here's this big, long, complicated, essentially how we do the Hardy-Weinberg principle. We're going to do a lot of this in class. So don't freak out. Don't try to learn this all tonight. But just kind of look through this slide here and see if you can't get a little bit of a grasp for what is being asked. Essentially, what we see is the formula is p squared equal plus 2pq plus q squared equals 1. And essentially what that's doing is taking the frequency of the homozygous dominant, the heterozygous and the homozygous recessive, and that should give us a total out of 100%. So we're going to look in depth how to do this. Don't freak out, but go ahead, look kind of through this and see if you can kind of see how it's done. You may need a calculator, so it will not hurt to bring a calculator. In fact, for the video quiz, what I want you to do, your secret word is calculator. Okay? That way you can hopefully remember to bring one as well. So, Hardy-Weinberg predictions can be disruptive. And the way they can be disruptive is if we see the genetic equilibrium broken. And there are 5 things below that show you how genetic equilibrium will be stopped if it's thrown out of equilibrium. And those 5 things are non-random mating, small population size, immigration or immigration, mutations, and natural selection. So the first we're going to talk about is non-random mating. And for genetic equilibrium to occur, individuals must mate with other individuals at random. Okay? So when we look at most species out there, even us as humans, we mate based on heritable traits such as size, strength, or coloration. Some species even mate based on the songs that they give off, or other interesting rituals. So this is called sexual selection. And when sexual selection is seen in a species, then that means equilibrium cannot work, because there is a selection based on traits. The second one, if there's a small population size, genetic drift is more likely to occur. If you have a large population, there's less of a chance for it to occur, and more of a chance for equilibrium, because you have more species. If something were to happen, there's not going to be a lot of shift. But if you have a small population, that's when you start seeing changes occur more frequently. So since there will be evolutionary change in the population, when we talk about a small population, it cannot remain at equilibrium. Then we have immigration or emigration. Individuals that come to an already established population are going to bring in brand new alleles into that gene pool. When that happens, there's going to be movement in and out of that population that causes some shift in allele frequencies. And we've been talking, if there's a shift in allele frequencies, there's not going to be equilibrium because that gene pool is being changed. Then we have mutation and natural selection. And we know mutations can introduce brand new alleles that have not been seen before. It can introduce new traits into a population, and it can definitely change the allele frequency. And that goes with natural selection too. If different genotypes have a better fitness than others, it's going to affect the allele frequency. So both of these, you guessed it, change the equilibrium. So all of those change the equilibrium. If we want to maintain equilibrium, all of those things we were just discussed cannot happen. So please make sure you write down any questions you have. Remember the secret word. Just so you all know, I will also have the day 33 video posted so you can watch that, get a head start, so you can have everything done before you go on spring break. Hope you have a wonderful day. Go out, relax, enjoy the weather, and I will see you soon. Goodbye.